Anton Bushuiev from Czech Technical University. Anton graduated from, got his master degree last year in Czech Technical University and started his PhD program last year. And he works under in the research group under the supervision of Professor Josef Šivec. And I'm very, very glad to welcome Anton with a very nice presentation on related to biology and many other interesting things. So you have 20 minutes plus 5 minutes for questions. We are slightly behind the schedule, but you have your time. Please, the floor is yours.
Доброго дня. Can you hear me?
Yes, everything is.
Yeah, perfect. And can you see my screen?
Yes, everything is perfect.
Yes, thank you very much for the introduction. Yeah, so my name is Anton, and originally I'm from Ukraine, and for the last 7 years I've been living in Prague, where I got my bachelor's and master's degree. Yeah, and it was mentioned I'm now a first-year PhD student supervised by Dr. Josef Šivec at the Czech Institute of Informatics, Robotics, and Cybernetics. And today I'm going to present our paper, which was published at ICLR 2024. And this is the result of very nice collaboration between our institute, Czech Technical University, and also Masaryk University and the Institute of Organic Chemistry and Biochemistry in Prague. And it's going to be a bit different from the previous talk because it's related to machine learning applied to biology. And as you can see, my presentation is going to be related to protein-protein interactions. So I want first to start with what they actually are. So this very nice animation shows how coronavirus attacks a human cell. So we can see that coronavirus has these spikes. So which are
visualized. Anton, we don't see new slide. If you have changed it. Yeah, no, it works.
Yes, sir. Yes, so in this animation, we can see coronavirus and how it attacks a human cell. And so coronavirus has these spikes. As we can see that they're visualized here in orange. And in fact, each of the spikes consists of 3 proteins kind of glued together. And we can see in this video another type of proteins, which are visualized in purple and located on the surface of a human cell. So these are receptors. And as we can see, coronavirus uses its proteins in the spikes to attach or to connect to these receptors, which enables it to get inside of the cell. And another type of protein, which you can see here, are antibodies visualized in blue. And we can see that they block the spikes of coronavirus, so preventing the attack. So we can see that this is some kind of a process which is abundant with proteins and their interactions. And this is not some cherry-picked example of a system where a lot of interactions between proteins happens, but in fact, protein-protein interactions underlie and the treatment and development of many other diseases, such as, for example, stroke, cancer, or Alzheimer's. And our collaborators at Masaryk University at Brno in Czechia, they focus on stroke. And as we can see, stroke is a medical condition when, as we can see on the right, stroke is a medical condition where a blood vessel is blocked due to the cluster of the proteins, which are called fibrins, visualized here in the beach. So our goal is to somehow break the clot of these fibrin proteins and restore normal blood circulation. And what we can do is to use a protein, which is called staphylokinase. And staphylokinase works in the following way. So we can take some dosage or concentration of staphylokinase proteins, put it into the bloodstream, and then the staphylokinase proteins start interacting with another proteins, which are called plasmids. And then they form what's called a complex. So further they operate as a single unit. And this complex or interaction of plasmin with staphylokinase enables to produce more and more of plasmin proteins on the surface of this clot, and which in turn enables to degrade or the fibrin. So essentially, as plasmin molecules, let's say, eat this fibrin clot and enable to restore normal blood flow. So this mechanism is very well studied for many years by our colleagues, but it's still not perfect. Specifically, staphylokinase is a very attractive thrombolytic drug candidate. It's known to be much cheaper and safer than state-of-the-art thrombolytics, but it has one issue. So one hypothesis is that staphylokinase doesn't kind of want to interact with plasmin as much as we want, which limits its activity. So our goal is to somehow make staphylokinase more attracted to plasmin, so to make it interact much more productively with plasmin, which may enable more effective thrombolysis. And so this question is how to make staphylokinase kind of more attracted to plasmin was initial motivation for our research. And before I'm going to proceed with our method, I just want to firstly say a bit more about proteins. So what each object in this slide actually means. So if you take, for example, the staphylokinase protein visualized here in blue and take a more detailed look at it. So it's again visualized here on the right. And we can see that proteins are in fact large molecules, or as they're called, macromolecules. So molecules consist of atoms and some chemical bonds between them. But what is very special about proteins is that this is not some random cloud of atoms connected with chemical bonds, but in fact, proteins have some very special structure. So specifically, proteins are sequences of amino acid building blocks. So all of this molecule consists of 20 possible building blocks, which are called amino acids, which is somehow folded in 3D. So for example, one amino acid is shown here, which is called phenylalanine. And since protein is a sequence folded in 3D, it's also often visualized like this, where we can see common folding patterns. And now, if we get back again to our question, how to make staphylokinase more attracted to plasmin. So, and if we take a closer look here, so again here on the left we can see the staphylokinase protein, which is an attractive thrombolytic drug candidate, and here we can see another protein which is plasmin from human blood. And if we take a closer look at what is called interaction interface, so where actually proteins touch each other, where the interaction occurs, we can see that the interaction is given by specific properties of this amino acid building blocks. So now, how can we approach this question? How to make staphylokinase more attracted to plasmin? So what we can actually do is to substitute or to play with different variations of these building blocks. So we can substitute some amino acids for another. And technically speaking, these substitutions are called mutations. And so we can try different variations of this interface or to introduce different mutations. But obviously, this is a combinatorial problem. And even if we focus only on these 3 amino acids, it's already 20 to the power of 3 combinations of different mutants. And testing even one in the lab may take several months. So it motivates to apply machine learning to predict what mutations or substitutions of this amino acid building blocks are favorable for the interaction. So before I proceed, so that I don't keep saying attractiveness or like how to make staphylokinase more attracted to plasmin, technically speaking, attractiveness is quantified by what is called binding energy or Gibbs free energy delta G. And here, so it represents the difference between 2 molecules in the unbound state or when they are unconnected and when they are connected. So we want to minimize this quantity. The lower, the better. So the lower the binding energy or delta G is, the higher the better or tighter interaction is. So it's exactly what we want to minimize with machine learning. And now it finally brings us to the, let's say, a bit more formal definition of our problems that we are trying to address with machine learning. So specifically delta G prediction. So before I explained what delta G means, but now, so the input for our problem is some protein-protein interaction. So for example, staphylokinase plasmin interaction in this case. And let's say we want to introduce some mutation here, so to substitute amino acid 34, and the original amino acid type was serine, and let's say we want to mutate it to glycine. And so we are questioning, so what we want to know is whether it's going to improve the interaction or not. So what we want to predict is how this binding energy or delta G changes. So that's why what we want to predict is delta G, so difference between delta G or binding energies. And again, we want to minimize this quantity. So here, since this is a difference between mutated and original interaction, if the mutated interaction is better, we will get a negative value of delta G. So again, we want to minimize it. And what is very important if we look at the distribution of delta G values, so in nature, so let's say if you introduce some random mutations to a protein, and we just measure delta delta G, we can see that most of the mutations are in fact bad candidates. So random mutations do not do anything good for us. They only disrupt the interaction. So that's why it's a bit like a needle in a haystack problem. We want to identify mutations which have negative delta G, and there are not so many of them. So to summarize, our goal is to take some protein-protein interaction and the mutation we're interested in and predict how good mutation is or its effect on the interaction quantified by delta G. And what is very good for us is that there exist datasets exactly in this form. So one dataset is called SCAMPI, and here, for example, we can see examples of 2 entries from the dataset. We have some protein-protein interaction, let's say 3 proteins interacting together. And then we know that in protein C, let's say this one, if we mutate amino acid 21 from cysteine in tubulin, let's say it's here, and we know that delta G is slightly negative, so which means that mutation is kind of a bit favorable for us. And then we have another example where delta G is bad, so it's very positive. So this data is perfect just to train some machine learning to predict, to approach the tasks that I defined. But the problem is that this data is very scarce. So in this dataset, which is quite comprehensive, we only have 300 interactions and 7,000 mutations are associated with them. But obviously, for such a combinatorial problem, that's not enough. So our main idea is of the paper is to break down this limitation by inspiring by the state-of-the-art techniques from natural language processing and computer vision, and specifically self-supervised learning from large unannotated data. So that's why in the first step, we constructed a large unlabeled dataset, which contains protein-protein interactions, but without any annotated mutations. And as we can see, it's several orders of magnitude bigger than this annotated dataset. And as I'm going to show in a second, from this, we can construct millions of training examples as opposed to several thousands. So how can we actually leverage this dataset for delta G prediction? So our main inspiration is masked language modeling. So a technique which, for example, underlines ChatGPT or other successful models in machine learning. So the main idea is that we have, let's say, some sentence. So what turns out to be very effective is if we take a lot of unannotated sentences and then we construct millions or billions of examples where we just mask some random words and train a model to predict them. And so it leads to very powerful representations of text. And so the question is, can we adapt this to our problem of predicting delta G for protein interactions? And the answer is yes. And it's shown in this slide. So first, the slide shows our self-supervised pre-training from the large data set we collected. So on the input, we have some protein-protein interaction sample from our data set. And then in the next step, we convert it to some graph representation, which is convenient for machine learning. So specifically here, one node represents one amino acid. And arrows here also capture orientations of residues, so where they kind of, orientations of amino acids, so where they kind of look. And then we connect all of these nodes with some 10 nearest neighbors so that we get a graph representation. And what is very important, then during training, we mask some of the amino acids randomly. So let's say 2 amino acid building blocks from here, visualized in gray. And then we apply some kind of larger neural network, which is a transformer also adapted to this 3D graphs. And then we get some hidden representations for each of amino acids or embeddings. And then as it's done in natural language processing, we simply classify these 2 masked amino acids onto the 20 possible types to predict. So as a result, we get output probabilities for these 2 masked positions. And then we optimize this negative log likelihood loss just to maximize the probability of the true or amino acids which are present here that we masked. And so in this way, we can obtain very rich representations of protein-protein interactions, similar to natural language or masked modeling in computer vision. But what I think is very interesting here is that this masked modeling is even probably more attractive for proteins than for, let's say, text. Because here we can easily adapt it to predict delta G or predict effects of mutations. because masking is kind of similar in nature to mutations or substitution of individual building blocks. So as a result, we can quite easily adapt this. So after the self-supervised pre-training, we can quite easily adapt this model to predict effects of mutations. So now on the input, we do not only have some protein-protein interaction, but we also know some mutation. So we know that so we have some set M of amino acids that we are mutating to some other amino acid types. And so what we do now is during for inference, we do not mask some randomly selected amino acids, but we mask the ones we are interested in mutating. And then we predict the probabilities similarly as it was done during pre-training. And then intuitively speaking, if we can see that the model predicts higher probability for the mutated amino acid types than for the original ones, from this we can kind of deduce that probably mutation is favorable for us. And this is here formally specified via this what's called log odds ratio. So here on the right, we can see a joint probability of the mutated structure, and on the left, we can see a joint probability of the original one. And if we take the difference, we can predict something which is reasonable to consider as delta delta G or effect of mutation. And then we can also fine-tune this pipeline using this small data set of annotated delta delta G data. So now I want to show the main results of our work. So now again, if we take this small data set of annotated mutations, we can use our model to predict delta delta G. Sorry. And the most important result is that our model called PPI former achieves state of the art on this data as compared to several other diverse representatives for predicting delta delta G. And nevertheless, it still doesn't outperform a very slow yet powerful simulation of effects of mutations, which, however, takes several orders of magnitude more time and which is not applicable in practice for our case. And then just to validate that our model also works in practical scenarios and not just on some standard benchmark, we took some data set of mutations associated with staphylokinase thrombolytic drug candidate, which I introduced earlier. So in this case, we have a data set of 80 mutations of staphylokinase protein, which is shown here. So, and we have 80 mutations for it, and 6 of them are, let's say, very good. So they improve the thrombolytic activity of staphylokinase a lot. And 14 of them are just good, so they improve it a bit. And 16, or most of mutations are actually disruptive, so they have a negative effect on thrombolysis. And here what we do is that we score all of these 80 mutations with our model. So we predict delta delta G for each of them, and then we sort them according to delta delta G. And then we measure if our model can accurately retrieve this good or very good mutations from some top K predictions. And here we have the ranks, so lower the better. And we can see that our model actually predicts 2 of these very good mutations at first 2 positions. So which shows the potential of our model to be applied in practice. If we screen some space of mutations, then we can select some top K and use them to validate in a lab. So to have some real experimental results. And then we also used another case study, so where we have a coronavirus spike here and some human antibody. And here we have similar observations from the pool of 500 mutations. We know that 5 of them are good, and we can see that our model actually gives low ranks for these mutations, which also demonstrates the potential to apply our model to predict mutations which improve interactions of antibodies with viruses. And in the final step, we made our PPI former model easily accessible for domain experts, also for biologists which want to find mutations which are good for their case. So we implemented this website where a user can provide some protein-protein interaction and the list of mutations here. And the website will return delta delta G predictions for all the specified mutations, and also show some visualization with some sort of explainability. So here, for example, is shown for this mutation where we substitute 2 amino acid building blocks, and they will visualized in red. And then it also shows the surrounding where the brighter some other amino acids are, the more important they were for the prediction. And this is the end of my slide. And now we are collaborating with Masaryk University with the goal of improving the staphylokinase thrombolytic. So here we can see some photo of real mutants of staphylokinase, where in each box there is one mutant. So staphylokinase proteins with some amino acids substituted. And we all are keeping our fingers crossed that we can reach our goal just to make a better drug for stroke. And so thank you very much for attention. And if you have any questions or ideas for collaboration, please feel free to reach us.
Thank you very much, Anton, for this insightful presentation. Are there any questions? Yes.
Thank you very much for your talk. And my question is, have you tried to apply any graph neural networks for your task? Because it's similar that in this biology domain, they are quite dominant now, maybe some graph convolutional networks or so, because you actually have some graph representation here.
Yeah, you're absolutely right. So in fact, Equiformer can also be considered a graph neural network, and we also try to apply other ones. So this, what architecture we are using here is in fact a graph neural network, but also somewhat combined with transformer. So yes, we are mainly using the graph neural network here.
Thank you. Any other questions? How generic is the approach? Is there anything special about these interactions that in the instances you studied, or do you believe that this is a general approach and it will work in any protein interaction prediction?
Yeah, so our model was developed to be general for any kind of protein interactions. So in our data set, which I showed here, so we constructed in the way that we mined all the protein interactions that are kind of available publicly to humanity. So we extracted just in like all kinds of protein interactions and trained our model on them. So our model was developed to be as general as possible for different sorts of protein interactions.
Thank you. Thank you very much. Any other question? If not, then let's thank the speaker once again. Thank you.
Yes, thank you very much for this.
And we'll be glad to see you, welcome you, Anton, some day in Lviv.
Yeah, I'm looking forward.
Thank you.